Okay, so we're going to go into the muscular system next. And basically, when we're here, we're going to go over um, the functions of the skeletal system, the anatomy, what we're talking about with skeletal muscle, how it controls muscle fiber contraction, the mechanics of how it works, how we produce energy, both anaerobic and aerobic. Um, Talk about the different types of muscle out there, the anatomy, and then go over how it integrates with some of the other systems. So, when we're talking about the functions, basically, a primary purpose of it is basically going to be through support and movement of the bones in the skeletal system themselves. So, our skeletal muscles are our main muscles that we are basically controlling or our voluntary control. Um, so, we'll talk about each of those. We're going to use that to basically produce the movement. It maintains our posture, body position. It's going to be supporting our soft tissues. This also does cover up all the fossas and foramen, and then helps us with our regulation of our body temperature. So those are all big major functions that I want you guys to be aware of and know. And they're going to be doing this by moving the bone and how it attaches. Some muscle is going to be attaching to the bone, and some of it's going to be through tendons, some's going to be direct insertion. It depends on what that muscle is going to be doing. We have some muscles which are fine motor controls. We're dealing with our fingers, and some that are basically less coordinated, needs less innervation, like our back muscles, which are going to be major muscle groups. So it all depends on what the muscle is doing and how it's going to work. It maintains our body posture, and it basically is able to move our system and allow us to move the skeletal system we talked about. So it's also going to support our soft tissues. There's our voluntary muscles is also going to be moving through for digestion process, going to be around our visceral organs. We also have it across our area for protection and support. All bunch of functions on there. And then a lot of it's going to be too, we'll get into, is maintaining and helping to maintain with body temperature, shivering, other aspects, and then as well as help us with release of body temperatures when we're getting too hot. So we'll talk about the anatomy of the skeletal muscle. And this is going to be another concept that I just want you to know here for the test coming up here. So gross anatomy, each skeletal muscle is going to be bound and going to be grouped together. But each of these is composed of a single muscle cell or single muscle fiber. So you can see this broken down of the skeletal muscle itself. And then we have little muscle fascicles, which are bundles of cell. And then our basic portion of the skeletal muscle fiber is going to be that single strand that you see there. So as we come out here, we have our different layers. We have our epimysium, which is going to be an outside, our paramecium, which is going to be surrounding here, and our endomysium, which is going to be surrounding our single skeletal muscle fiber. So those are our three layers that are going to be around that. The epimysium is basically going to be the most external layer. Epi being on the outside, endo is usually going to be our inside one. And that's kind of where I'll leave on those. When we're looking for here, basically these are all organized together, and then at the very ends of the muscle usually connect to tendons, and tendons are what actually connect the skeletal muscles to the bones. So as these muscle fibers come down, right in here you can kind of see interwoven in here, the tendon attaches to these muscle cells, and then that tendon is what's going to attach onto the bone, and that's what allows that motion and movement there. And that area that caused that binding is called an aponeurosis, and that's the collagen fibers that attach the skeletal muscle to the tendons and then also around to each other. So inside this area, we got to have pathways. So the epimysium, same as we talked about in the bone earlier, and the paramecium have pathways for blood vessels to get in. The blood vessels are going to come down, and that's going to be important in feeding it and providing it energy. And then it also is going to help us with getting rid of the waste products as well. Because as we come in, as you're aware of lifting and that, you get lactic acid buildup as a byproduct, and we'll talk about that later. So we need to clear that away as our waste products as well. So we got to get oxygen and nutrients to it, and then we got to be able to remove the waste. Also in there, to know when to move, we have to have our nerves going in there. So the axons will go down and they'll penetrate into each of these muscle fascicles, and that's what's going to start an action potential and allow those muscles to contract or relax based on whatever we're needing to do. Most of the skeletal muscles are very large, and our skeletal system is a big difference between that and the smooth muscles and then the cardiac is that these are multinucleated. There's hundreds of nuclei in there, which basically help control the motion, and because we want them to work, they need to have that ability to work and focus. So as we look here, and we're looking at that muscle fiber, and we get down to that single muscle cell, we can even break that down in even further area. And this is that single muscle cell I showed you before, and I'm going to go back to that photo. So when we're looking here, this single muscle fiber right here can be broken down into this here. So through this muscle fiber, we have this sarcolemma, which is kind of the covering around here. And this allows where our vessels come down in and get to each of the tiny cells or portion in here, the filaments that make that area. And up here we have basically what's called a thick filament and a thin filament that work together. It's surrounded by this terminal cistern or the sarcoplasmic reticular around each of these little fascicles or little bundles here. And then inside here as well as the T-tubules, which allow the nutrients, blood vessels, and that to be flowing in there. And then there's mitochondria and other portions of the cell that are around there that's going to be help with producing energy. So this is all a function of how it's going to be moved. So the sarcolemma, again, has these tubules, and these T-tubules is where we get a passage into these muscle fibers, and that's where we get different chemicals that are going to be released in here. We'll talk about calcium, we'll talk about all those motions, and allows for the contraction. So what we have here is we have a thick filament, as I'm showing right here, and these thin filaments. And this band right in here is going to be the muscle fiber. And this is what actually is the one of the smallest aspects of this myofibril. And this is where this contraction occurs. And contraction occurs in this area right here I'm showing you. And then that happens throughout the entire muscle belly, throughout areas. And this is what allows shortening and lengthening. And when it's in its most relaxed state, like this is showing here, you just get a small area where the thin and the thick filaments are overlapping, but they're basically a big space in between. When these contract, you'll see these will overlap very close, and they can shorten quite a bit. And there's hundreds of these myofibrils in each basically bundle. And then what we do is we get these thick and thin myofilaments to contract and move on itself. That's basically with the proteins that are coming to, and I'm going to talk about that coming up, about these actin proteins and these myosin proteins. Now the actins are in the thin filaments, which is more the fixed side, and the myosin is the thick filament, and that's that floating area that kind of contracts and moves within that ligament, within that muscle fibril. And then the sarcoplasmic reticulum is this portion here, and that's the surrounding area, and that allows the T-tubules coming in, and we'll talk about that contraction here. So let's go back and talk about this thin and thick filament. So here is the myofibril, and on that myofibril, this portion right here, this main small area, is what's called the sarcomere. This is the smallest portion, or basically aspect of the myofibril, and where that basically extends is from where the attachment of the thin filament is on one side to the attachment of the thin filament on the other side. And this area right here is called the Z-line. The Z-line part of the myofibril is the attachment of that thin filament. That's not moving to this portion. Then what we have here is we have an I-band. An I-band is basically the portion from the thin filament to the thin filament with that connection of the Z-line. The M line is basically going to be the exact opposite. It's going to be from the area of the where the thick filament attaches, and the A band is going to be from the length of one thick filament to the length of the other thick filament. And then this is that overlapping zone that I talked to you about. So when we see the contraction, you'll see this portion of the thick filament can extend all the way down to here, and that causes shortening, as well as this one all the way down here. So that area, as it contracts, will basically shorten quite a bit. And this is that overlap that we're seeing. And then that resting length is usually about 2 micrometers. The thick filaments are in the center, as we talked about, and here we go back to that picture. And so this picture right here, I want you guys to know, in case you were to see something like this again. I want you to be able to know which ones are the thick filaments, which ones are the thin, the Z line, the M line, I band, and A band, and that's all part of the sarcomere. And we're going to talk about how that contracts and how that moves. And here again, as we're looking at it through another picture, and this is where it shows up. And again, this is that thicker filament, so you can see this is going to be a darker and lighter, and that's what we'll see on an electron microscan look right there. So what happens is the thick filaments have this molecule of myosin, and the thin filaments have the actin molecules in it. So here we're looking at the thin filament, and you can see this is all kind of bound up, and we have troponin and tropomyosin. That's this band that comes down here, and then we have all these little actin myosins. This almost looks like a double helix that we're looking at, and this is our thin filament, and that's how that looks. What'll happen is these proteins will set in position, and we need to basically open these up so that the thin filament can interact with the thick filament, and that's what's going to get us our muscle movement. Well, how does that happen? Well, our thick filaments I told you about had these myosin molecules, and you've got this large myosin head which is able to rotate on this hinge, and here's your myosin molecule. So your thick filaments have all these different myosin heads and these myosin molecules that go in, and this is what's going to interact with those actin that we talked about. So what'll happen is, here's that myosin head right here, and we have ADP that's coming in here, adenosine diphosphate with another phosphate molecules. When we get release of calcium into the system, and I'm going to talk about that more when we get in the nerve, but as calcium comes in, calcium will bind with that troponin, and that causes a shift or a movement of this thin filament to open up, and it exposes those actin cells. When those actin cells get exposed, that's going to allow binding between these myosin head. So as we look here, calcium opens this up, and you can see calcium binds to the troponin, opens up, so now we have access to these actin molecules. Now, with the use of energy, basically breaking off using an ATP or an energy molecule, the myosin head attaches. And as it attaches and moves, you can see it flexes, and it basically pulls that thick filament along the thin filament, and that's what gets us contraction. And here's the next phase. So now it's contracted, it's moved along. Now we've basically used up our ATP, and it releases the head and the area relaxes. So this is where we use energy to basically cause this motion of the muscles and that sliding factor. So when we start losing up our energy area, then that releases, the muscle slides back to the way it is. So we want to talk about the control of the muscle fiber contraction and how we do that. Well, basically, because this is a voluntary muscle, this is all done under our nerves. And basically through, as I go through the neurology lecture, we'll talk about getting that signal down to the muscles, which causes a release of calcium into there, which allows it when to contract, and then also pull that calcium out and release energy and stops those contraction as well. So that's going to be a nerve impulse that's going to be controlling all this, and we'll hit that more in the neuro lecture. But that neuromuscular junction is basically where the nerve comes in and attaches to that skeletal muscle, and here's a look at that right now. So here are your myofibrils that we just talked about, the mitochondria in there, and that synaptic terminal is right where that nerve comes down in here, and it attaches to this myofibril. And this is what controls, and we get that action potential or that electrical impulse, whether to contract or stop contracting, is going to come down, and this is where that communication is going to happen. How this happens is through a neuromuscular neurotransmitter. Acetylcholine is going to be our main one that we talk about as we release acetylcholine into this area. That's going to go across and bind to the sites on the muscle, and it's going to allow our impulse to travel into the muscle so we can get our contraction and reaction. So here's a close-up look. So right here we're seeing that synaptic terminal. Here's a close-up look right here. So here's acetylcholine, and it's all in this area. And when an action potential or a nerve signal comes down, it causes the release of this acetylcholine to come up and attach over to the sarcoplasmic reticulum and open up these little channels. As it attaches here, it opens up. And that's what allows our motion or contraction to occur. Now again, this is acetylcholine, and then it can actually be broken down by what's called acetylcholine esterase, and I'll get into that over in a little bit later. But basically, the release of the acetylcholine comes across these terminals, attaches, as you can see here, at the synaptic cleft, and as we pop right over into here, opens up these channels and allows sodium to come into these channels. So they allow that to diffuse, and the permeability of the sodium ions, and they rush into there, and that helps to continue with that action potential. So here you can see, to stop that action potential, right here we have to get some binding with that acetylcholine, and that stops it from attaching, and it breaks everything up, and the action potential then gets stopped and closes up. But when we get this initial attachment, that allows the sodium into the channels, causes a sudden trigger of that action potential, and then the sarcoplasmic reticulum does a massive release of the calcium ions through the terminal cistern into those myofibrils. And again, I talked about that calcium is going to bind with the troponin, allow a twisting, and allow those actin-myosin molecules to be opened up. So here is a diagram showing how exactly all that works. So I'm going to start over again. Nerve impulse traveling down to the synaptic terminal. The synaptic terminal releases acetylcholine, binds to these receptor sites right here, and as that does, it causes an influx of sodium, which potentiates the action of the muscle, the synaptic action potential, and it comes right down through the myofibril into the T-tubule. Once it hits this in the sarcoplasmic reticulum, that releases calcium, as you can see, into the myofibril. Calcium attaches to this troponin, causes shifting of that troponin to twist and allow access of the actin. Once those actin is exposed, the myosin head attaches, and with the use of energy, causes contraction. And you can see right here, you'll see that contraction will start, and that thick filament will start sliding. These are all the little myosin heads. Will start sliding or shortening, and that gets contraction. And here's another example showing that. And you can see how much that shortening can occur with each cross bridge as it comes over. Once we've used up all the energy in the area, it will relax. The head will release, and the thick filament will be able to slide right back down. This is all those steps we just talked about. There's a release right there. The thick filament will slide back into the position it was prior to start, and we go right back to the beginning. And how this happens is our acetylcholine is removed by acetylcholine esterase. This is another chemical there that basically binds with the acetylcholine and stops it from binding with the synaptic terminals. It stops the influx of sodium, and therefore then the calcium gets all reabsorbed back into the sarcoplasmic reticulum, causing shifting of the troponin head, and the troponin molecules. It locks up that actin again, slides back into position, and there we get contraction stops, and we get relaxation occurs, and those muscles slide, those fibers slide back into position, and we get lengthening. So that's basically the contraction cycle. Now, why is this important? Well, this is important for a lot of different reasons. We're going to talk about acetylcholine and those nerve transmitters, and so when we start talking about our RSI, and other drugs, we're going to talk about how that works to stop the muscles from moving. And this is kind of one of those areas. We're also going to talk about the need for sodium. We're also going to talk about the need for calcium and how that helps with contraction and what happens within the body, especially also during, we'll talk about rigor mortis and how that happens, where there's a release of calcium in the area and we get a strong binding. So all these chemicals that we're going to be using and different drugs that we can use are all going to play a big important factor, and you really need to know all this anatomy physiology. So I'll make sure I'll be going over this again later in different lectures, but I want you to understand how this process works, because it's kind of the basis of why medications work, why we're using them, and helps you think through those processes. All right, we'll talk about muscle mechanics in the next lecture.